Hello, everyone. Welcome to today's session. So we will be starting here in few more minutes, and we are also going to be streaming this meeting live. So if anybody feels like, you can also watch this session live on YouTube right now. And we are going to be starting at 6:30 p.m. IST. So thank you all for joining today's session. Hello, everyone. So we are going to be starting here in 5 more minutes. So if you have any questions while we are doing this session, please feel free to post those questions in the chat box. We have team present from each of the collaborative partners who are doing this data science and drug discovery program together. And I would be introducing you to everybody very soon. So please feel free to post all of your questions in the chat box. And today our mentors would be addressing those questions, and our team will also be taking in those questions. So thank you again for joining in today.
Boleh.
Good evening, everyone, and welcome to today's session. Today, we will speak about the upcoming program on data science and drug discovery. This program will introduce participants to the application of data science to important challenges in clinical and pharmaceutical applications using practical examples that cover genomic, transcriptomic, and structural data. This is a collaborative program and conduct and organized by FABA Academy, which is the Federation of Asian Biotech Association, Pharmatoppers, Pine Biotech USA, and Pro Biotechnica Research Private Limited. Let me now introduce you to the program mentors. We have Dr. Harpreet Kaur, who is a certified OMICS logic trainer and expert bioinformatician. She will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is the OMICS Logic project mentor and an expert in big data computational analysis. He has a PhD in computational biology and over 10 years of experience working with industry and academia. Dr. Sujit Tangar Paliwar is the founder of Pharmatoppers. He has a PhD in machine learning, chemoinformatics, and natural language processing. Ilya Brodsky is a co-founder and CEO of Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners who are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Ms. Neetu Singh is the CEO and founder of Pro Biotechnica Research Private Limited and specializes in life science research and development. She's a gold medalist in MTech Biotechnology from Amity University. My name is Beepsha Biswas, and I am the OMICS Logic Community Manager, responsible for daily interaction with thousands of users who are part of our bioinformatics community managed by Pine Biotech. For those of you who are joining us for the first time, I wanted to share about the OMICS Logic programs that are a result of a close collaboration between Pine Biotech, an innovative big data analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the OMICS Logic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. OMICS Logic is an international program that is running in 5 different regions with over 10,000 users around the world. Due to this fast growth, our team is working with local and regional coordinators who are helping refine the local program logistics and leveraging our online training resources, adapting them to the needs of the students and researchers around the world. In this OMICS Logic programs, we combine online sessions like we are doing today, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in the well-known and respected OMICS Logic programs. So what are the outcomes for a student who attends one of these OMICS Logic programs? These outcomes are as follows. A student would learn to read and understand bioinformatics literature, perform basic NGS data analysis processing steps, analyze data and form hypotheses, prepare data for machine learning, cross-check signals with gene databases, know what type of data is required to answer scientific questions, create your own bioinformatics analysis, and present the results in an organized way. All of these skills are essential in bioinformatics and can also be translated to other fields. Now, I would like to invite Ilya Brodsky, our CEO at Pine Biotech, to talk more about the upcoming program. Over to you, Ilya. Thank you.
Thank you, Beepsa, and good morning, everyone. So good morning, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. Our vision is to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and expert support. And you can see a little bit more about what we do at Pine Biotech on our website, pine-biotech.com. Our mission is essentially to simplify bioinformatics or data science and advance research through our modular and intuitive multi-omics analysis platform that is powered by both human experience and artificial intelligence. We offer training, research support, and big data analytics tools that are designed for research and biomedical discovery. Our mission is to support cutting-edge research, but also help many users solve big data challenges by developing innovative solutions with our partners that are easy to use, providing training like this program that will enable users to use these tools effectively and to help scientists arrive at sound and reproducible results based on data that they generate. So in my part of the webinar, I will share with you a little bit of an industry perspective on the growing demand for data science skills and training in life sciences. So I will briefly talk about my perspective on how such skills can be utilized in various projects that I have been exposed to over the past few years, and provide a perspective on how this program can help you gain an understanding about the various domains within bio and chemoinformatics, preparing you for industry-specific or research-specific work and projects in your future career. So let me start by talking a little bit about why omics and why genomic data has become so central in life sciences. So after the discovery of structure and function of the DNA molecule in the 1950s, that was about 70 years ago, humans have been on a tremendous worldwide effort to really discover the meaning and function of DNA code. And it took almost 50 years from that time to sequence the first human genome, but soon after that, the technology developed during that time started evolving rapidly. So by 2015, the 1000 Genome Project brought in a significant number of whole genome sequences to the research community. And in 2018, over 100,000 whole genomes were already sequenced. Today, there's an ever-increasing number of genomic, transcriptomic, proteomic, and metagenomic datasets that are available to describe subcellular processes of regulation with highly detailed digital data. And so as a result, we really live in an era of omics data. So this is data like genomic, where we talk about mutations, gene expression patterns in association with clinical data that can help identify molecular biomarkers for diagnostics and therapy selection, as well as an abundance of data that allows for novel discoveries that provide us with a better understanding of complex diseases at the subcellular molecular level and help us identify targets for novel drug design and drug repurposing. And so what we see is that there's a critical need to train all levels of life science research and support in dealing with this data. And so because at the center of data generation, data analysis, and decision making stand people that need to understand the logic of data analysis. So they need to know in what ways to improve data collection and what kind of data is needed to perform the analysis. They need to understand what methods are needed and how these various heterogeneous data sets can be integrated, and how the analysis methods can be useful in actual decision making. What can be trusted, and where do we expect model overfitting or lack of representation? So really, the skills of data management and analysis are needed across this whole spectrum of applications in life science and industry. And so that's why our company has developed the OMICS Logic program, which are designed to provide training in bioinformatics, enabling independent research that is guided by mentors who have such experience, but also through peer example. And so what we are seeking to design is more than just a training program. It's a growing community that has a shared passion for data-driven research and appreciation of citizen science. And our goal is to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in data science. And so we hope that this program will be one of the examples where you will gain a clear perspective on the application of bioinformatics, machine learning in AI and research, as well as clinical, industry, and pharmaceutical applications. So to do that, we will provide you with access to tools and enabling infrastructure. We will expose you to the analysis logic for real data sets, how one in industry or research would apply such understanding to a real data set, and provide you some project examples where you could practice and develop your own skills. And so we've designed this methodology that allows you to understand the way projects are conducted by experts by giving you access to the same methods and big data analysis tools, and also opening up the perspective on how you can apply additional methods and variations to the same pipelines to a novel discovery. So this program is going to give you several key understandings about data science in research and industry. First of all, it's going to allow you to understand the details of molecular or biomedical data, which includes omics data, how this omics data can be understood in the context of what's happening within the cell, tissue, and the whole system. It will also provide you with an understanding of how the different interactions between the molecular features are significant for a phenotype, which is the collection of all expressed proteins in a given cell. And so then we'll talk about the specifics of next generation sequencing, which is one of the important data sets that allows us to study a variety of different signals that are at the subcellular level, understanding how to process, analyze, integrate, and interpret these molecular signals. We'll then turn to a section on data science, where we will introduce you to scripting practices in R and Python for statistical methods of analysis and machine learning. How can you load a data set and prepare it into an object? How do you actually analyze data using a variety of packages? How can those data sets be visualized? And what are the specific practices that you should follow to make your project reproducible? And then finally, we will introduce you to computational biology, where we will look at protein-protein interactions, ligand-protein interactions, and qualitative analysis of structure-activity relationships. So we will look at how you can apply the understanding of 3D structures and genomic function to understand drug repurposing and novel discovery. And so what this program will provide you with is a clear perspective on genomics in research, the application of processing tools for big data, where genomics, epigenomics, transcriptomics, and proteomics data are regularly processed on large server infrastructures and allow you to extract meaningful signal and structure data for downstream analysis. We'll also discuss different types of methods of RNA quantification, where we will look at microarray data and next generation sequencing and compare the different types of signals that allow us to extract meaningful and biologically interpretable information. Now, to give you a little bit of a perspective on where the industry is in terms of utilizing such data, I'd like to share just a few examples from my own experience where such data has been useful to understand specific types of applications and different types of industry and academic projects. So one of the things about omics data is that today you need access to tools that provide you with ability to understand patient information using animal models and oftentimes cell and organoid and tissue that could be modeling what happens in the real world. And so to do that, you have to understand how the different layers of data could be integrated, annotated, and modeled so that you can understand the real biology that takes place in an individual. And so one of the things that we see today is the need for data integration, where we can take information about the behavior or the activity of an animal model, where we can understand the genomic components as well as their structural applications and integrate that into a model that can describe a biological process. A lot of times such data could be used for biomarker discovery, where we would like to understand how a specific treatment is effective or ineffective for a group of patients. So we can do patient stratification, effectiveness of treatment, and an understanding of molecular drivers behind specific types of drug interactions with a pathology. We can also look at small molecule screening, where the understanding of the big data generated through high throughput experiments can help us select a specific focus library of molecules and apply downstream analysis such as docking to understand what will be an effective way to interact the ligand with the protein and test it in real life. And obviously today we live in a pandemic. So what's important is we will see how genomic data from such pandemics and different pathogen can help us interpret the different types of manifestations of this disease and try to understand how the genomic component drives the specific interaction between the pathogen and the immune system. Understanding how the interaction happens and translating our understanding to the structural components that have been characterized and will be important to understand in terms of their specific domains and the interactions with the host. We will also talk about some examples where you can appreciate the benefits of structural and functional analysis and annotation to understand how this impacts drug design as well as vaccine design. And so to facilitate this program, our team has been working hard to prepare training materials, data analysis tools, curated project data sets, and interactive sessions that are designed for this type of project-based learning. So if any of these project examples that I've described up until now sound interesting and something that you are interested in pursuing in your own research or career, I think that you would benefit from such training. And a lot of times it can be overwhelming. How do I actually get started? Because many of these projects include thousands of samples with large data sets. They include complex analytical tools that need to be combined together into complicated pipelines. And you need to be literate in different types of analysis methods, packages, visualization methods, and ways to deal with specific types of data. So I think that one of the benefits of this program is that even a novice who has limited experience in programming, bioinformatics pipelines, or big data analysis tools will be able to leverage these elements of the training program that we've designed to quickly get up to speed and be able to conduct independent research using some of these tools. And so how do these tools actually work? Well, one example of such a tool is a browser-based coding environment where you can learn coding in R and Python using specific libraries that are commonly used for data visualization, preparation, and analysis, where you will learn how to perform multivariate analysis of omics data using clustering and dimensionality reduction techniques, as well as looking at a variety of data mining and classification methods where you can train machine learning models to predict specific outcomes and identify relationships between significant features that you are informed about in your data. Now, these tutorials provide a very clear code breakdown. That code breakdown could actually be run inside the browser to produce the results and change those based on the input data sets. And so we've prepared a number of project examples that range from basic visualization to advanced analysis of metagenomic and transcriptomic and genomic data, including examples that really illustrate how this is applied in the world of pharmacogenomics, research, and applications to a variety of clinical and industry problems. So I'm excited to welcome you all to join this program on the data science and drug discovery, where training is going to be provided by real experts in the field, and you will gain support for a project or a specific type of a research question that you might want to ask in the context of clinical and industry applications. So I would now like to pass it on to Dr. Mohit Mazumdar to speak about the specifics of the program schedule. Mohit?
Hello, everyone. Am I audible now?
Yes.
Yes, Dr. Mohit.
Sorry about the interruption. Actually, the system I was using is not, the Zoom interrupted. So Biksha, can you share my slide deck, please?
Give me a moment, Dr. Mazumdar. I'll share your slide deck.
Sure, thank you. And hello, everyone. Welcome to this webinar, and I'll be talking in just a minute, and we'll be sharing my slides. Sorry for the interruption. So,
Dr. Mazumdar, you can continue while I start. Go ahead and share your screen.
yeah, okay, thank you. And I would like to thanks Ilya first for this detailed overview about the application of data science and its role in drug discovery. So this research-based training program is basically designed to help young researchers and students to take advantage of the bioinformatics resources for analysis of complex, high-throughput life science data, and also become versed in bioinformatics. So this program is basically designed for everyone without having prior knowledge of bioinformatics, but to having a basic knowledge of molecular biology and the topics that we discussed so far. So for everyone, my name is Mohit Mazumdar. Thank you, Bipsha. My name is Mohit Mazumdar, and I'm one of the global representatives of Pine Biotech. And my PhD was focused on developing a novel algorithm method to identify and predict calcium binding sites and study its binding affinity towards calcium. So during that process, I had to master data mining skill sets and become proficient at predictive analysis as well. In my current role, I am collaborating and working with academia and industry to provide support for bioinformatics education and research. Along with our hardworking and efficient team, we have been able to work with more than 30 universities for research-based programs like this, and we've been able to help thousands of students to develop understanding of data-driven research in life sciences. So today, I wanted to guide you all through this innovative research program, which is an outcome of a modern life science research, as you can see. So the application of machine learning for drug discovery and the application of genomics, microscopy, and quantum chemistry, they have like really created a radical impact, right? And that holds the potential also to significantly accelerate the process of this medical research and vaccine development, which is such a necessary thing, in this case that we are living through this pandemic, like COVID-19. So so before I talk more about the details uh, of these uh, about the details of the program, let's review this publication that you can see on the screen. So this publication is a recent one which could help us understand the importance of developing skill sets for the future and today's modern academic and industrial research. So why all of a sudden there has been an increase in application of data science and machine learning? So that's the question I'm asking. So also please feel free to write in the chat if you have any questions, if you have any doubts about whatever we are saying, and we'll be able to address those. So as I was telling you that why? So the reason is biomedical research is increasingly becoming a high dimensional science. So Ilya did talk about machine learning and the dimensionality problem, and like how we are using multiple types of data to answer a research question. So one of the challenge is the high dimensionality of this data. So especially in the pharmaceutical industry, now data is getting generated like really at a cheaper price. And because of that, not only in pharma but also in academic setup and in a research lab, so because of that there is a problem which is happening because of the increase in complexity and the diversity and the volume at which this data is growing. So it's really at a fast pace. So this is a result because of the development of these noble technologies that came for molecular profiling, imaging, and other types of high throughput readouts at an unprecedented rate. So how this is influencing the drug discovery pipeline, and why pharmaceutical and the industries and the labs are adopting this, and why we should learn these skill sets. And you can see that the author here describes how to incorporate successful data science to drug discovery pipeline, right? And that eventually cutting the cost dramatically and also reducing the time. So this has been implemented by most of the software industry, most of the pharmaceutical industry, and also small biotechs. Along with that, the academic setup where we are going for PhDs, when we are applying for master projects or masters, or we are applying for postdoc research. So this sort of research is being carried out now at an unprecedented rate, where people are expecting to gain insights from this big data, because this is something that have not been done before, that we've been able to generate at timeline data, where we can see the disease progression, where we are using machine learning. So I'll be talking about all of that. But before that, let's look at the maybe with Deepsha, we can go to the next slide and understand that how this course is actually meant to give you an overview of what is going on in modern setup and what is going on in the industry. So now let's look at the entire course curriculum and the units. So this special omics logic research, this research program is like it's a very unique opportunity for students and researchers to learn in depth and get guidance and support by academic and international industry experts. So in this program, the participants will work on example NGS and structural data sets, and they will be able to explore publicly available databases and resources to work on their independent and group projects. So this entire course will have 33 scheduled sessions, where the sessions will be divided by either online, like the online sessions, right, like we are having right now. Also, there will be coding sessions, and then going through the tutorials. You will have to be go through the tutorials that we will be sharing every week, before and after the session, as a part of the asynchronous learning. So this program will have a lot of components, and one of the major component is a strong feedback process. So you can see in there the highlighted parts and assessments. So they are feedback. That's the feedback process. So the feedback process actually helps us, and also helps the participants to make the most out of the resources. So, and that also helps the mentors of the program to evaluate and grade the participant progress. So there will be literature review, there will be other resources that we will be sharing with you. So in this program we will have several case studies that Ilya just talked about, and the case studies that we have prepared by working on, by working before the program and setting up and looking at the data sets, to help understand how you can access and understand the publicly available data sets, like on NCBI, or like on TCGA and other repositories. So now let's look at briefly about the entire course structure. So let's go to the next slide, Deepsha. Thank you. So after doing the registration, once you are registered for the program, so after that, the first week, till the first week of the program, when the program is going to start, the participants who register will have orientation and introduction sessions, where we will be collaboratively working with the participants to set up their academic profile and set up their accounts on big data analytical platform, TBioInfo platform. So this week and the time before that, we will be learning about big data and an application of bioinformatics. So getting prepared for the first week of the program, and the time before that, once you register, will be focused on getting you prepared for the program. So we will be able to help you understand what is big data and how bioinformatics is applied to these different problems. So the phase one, so once this introduction is complete and orientation is complete, the phase one of the program will be focused on the genomic data analysis for biomedical drug discovery. So here we will start with the basics. So we will start from where we will start from the basics of understanding of DNA sequencing, and using techniques such as sequence alignment and mapping the raw sequencing reads to the genome. So the first week will be starting of the genomics program, or genomics phase will be about understanding the basics and understanding about the techniques that are very much popular. So in the complete genomics phase for biomedical drug discovery, we will learn about NGS genomics, so Sanger genomics, exon, and we will look at the WGS and WES, and then we will also learn about the basics of DNA code and how variations can be learned. Like we will be learning about the mutations, we'll learning about the substitutions, and that would be also you will be able to also learn about those by looking at and by doing the courses that will be shared with you. So genomics 1, an introduction to genomics, and then DNA code and variation, then sequence alignment and analysis in genomics 2, and then looking at the GWAS and sequence and structure and function, which will be covered in the session. So in this genomic phase, we will also be working on projects. So after the genomic phase is over, in the second phase of the program, we will learn about the second data type, which is to understand the application of transcriptomics data in biomedical research. So in this phase, we will be learning about RNA sequencing. We will be learning about differential gene expression and machine learning. So in this phase, we will also be learning about how gene expression data can be utilized in several of these applications in several disease types. So we will start from how you can process the data. We will learn about mapping, we will learn about quantification, then we will learn about normalization, then we will learn about exploratory data analysis and differential gene expression, and then we will then we will then we'll move to machine learning. So how you can interpret the big data, that is the gene expression data, using machine learning techniques. So along with this regular webinars like we are having now, and the practical exercises that I talked about, and on then we will be also you will also be having access to the online courses. So these resources that you can see on the screen, they are tutorials with a complete curriculum. So each of these course covers an important aspect of transcriptomics data analysis. So when you go through these courses, you get an overview of processing, you get an overview of statistical analysis, you get an overview of machine learning tools, and you also get an overview of single cell data analysis techniques, which is a part of machine transcriptomics 4. So from 1 to 4, you kind of move from another one level to another. So these will be a part of the program, which will also have practical examples. So here, as we were discussing about the practical examples or the projects, so we already have prepared various examples you can pick from, ranging from biomedical to agricultural studies. So some of the projects have been developed for this program that have been earlier mentioned by Ilya. But also I would like to mention some of the projects that we have prepared includes modeling precision treatment of breast cancer. So that is one project. Drug repurposing of a drug resistance in liver cancer. So that is another project. Third project that we have prepared is drug discovery for Alzheimer's disease. And then we have a COVID-19 drug repurposing project as well. So this will be about the second phase, which is the transcriptomics phase. So we have completed so far 2 phases. First phase, we learned about the genomics. Second phase, we learned about the transcriptomics, and there will be a lot of sessions between these two phases. Then we will move into the third phase. So this is a 4-month program, and we will have you will have a lot of time to go through what I am describing for each of these sections, and you will have a lot of time to discuss with the mentor, and we'll be able to guide you through that process. So the third phase is about data science. So once you learn about the technology, now you will be able to analyze it further using machine learning techniques. So data science, as you know, has a potential to make an impact in all operational pipelines, whether it is in pharma, whether it is in business. So data science has a very critical role, and especially in pharma. Interestingly, data science has been making a lot of impact, for example, in optimizing clinical trial pipelines. There have been a lot of cost control. So the third phase of the program, we will learn about the machine learning to interpret and analyze biomedical data. And this will include like practicing on the coding platform like Ilya showed, and to learn how to perform clustering and data mining and predictive analysis using several of the machine learning algorithms that you can see on the screen. So yeah, so we will be using the ones that are being popularly used by the scientific community to answer and to find patterns in biological data. So along with these machine learning algorithms, Deepsha, if you can go to the next slide. We will also be utilizing the resources that we have developed for machine learning and AI that utilizes R and Python. So you will be learning about, so these are the resources that have been developed by Tauber Bioinformatics Research Center and the partners and collaborators of Pine Biotech. So we will be able to learn about how to load data and do visualization. We'll be working with sequences, and we will be doing, as I said, exploratory data analysis. We'll be utilizing statistics and machine learning in this entire phase of the program. So this entire expertise that you have developed by learning about the multi-omics data analysis, by putting that information and then applying machine learning. This could actually help us, or this could actually lead to new pathways. You can find new pathways, new targets that you can work upon for the next step. So the way the program has been designed is just like the pipeline that you show that you saw at the beginning, which is typically shown by a pharma industry, that how a drug discovery process takes time, that is like 10 to 15 years. So what we are trying to do here is that we are trying to understand couple of NGS data types, and then we are trying to understand the application of machine learning. Then we are coming to those identifying those genes, important proteins, by doing gene set pathway analysis or pathway enrichment analysis, which will be a part of the biological interpretation. So after this, we have the fourth phase. The fourth phase is about finding the target. So after you have found the target in the fourth phase of the program, we will learn and explore 3D structures or our target proteins or the chemical entities or the structural molecules. So to talk more about cheminformatics, I would like to invite our industry expert here today, Dr. Sujit. So over to you, Dr. Sujit, to help us, to guide us through the fourth phase and your expertise. Thank you. And please feel free to put it in the chat. I'll look into the chat and I'll answer if you have any questions related to the program and what we have covered so far. Thank you.
Thank you, Dr. Mohit. I would like to thank Ilya as well and Bipsha for this wonderful webinar in collaboration with Pharmatoppers, Pine Biotech, Tauber Institute, Probiotechnica, and FABA. Let me share my screen. Good evening, all of you. This is Dr. Sujit Tangadpallivar. I'm a founder of Pharmatoppers. Currently, I'm working as a lead data scientist at Arametrics, which is a subsidiary of Sun Pharmaceuticals, India. I have completed my PhD from NIPER, National Institute of Pharmaceutical Education and Research, Mohali. I have over 10 years of experience, industrial and academic experience in artificial intelligence, machine learning, natural language processing, computer vision, and cheminformatics area. I have developed several industrial usable softwares in the field of data science and chemoinformatics during my experience as a industrial lead data scientist and during my academic experience. I have published more than 13 research papers in peer-reviewed journals and conferences. So to introduce about our company, Pharmatoppers, we have around 20 plus experts in our team. We have 3 managers, 2 animators who animate those videos which are in YouTube. We have 10 advisors from abroad and many more. Team comprises many more than this. And we are also specialized in GPAT, NIPER, such kind of training, national level training program or a national level guidance program for the students, specifically for the pharma students. So along with the GPAT, NIPER, we also provide drug inspector, BITS, Pilani and Manipal entrance examination coaching. Along with that, we have several industry-oriented and skill development programs like clinical research management, drug regulatory affairs, data science, dry lab hands-on tools, specific topics, and so on. So in this collaboration with the Pine Bio, US, Pharmatoppers, India, Probiotechnica, and FABA, Federation of Asian Biotech Association, we have collaboratively launched Biomedical Data Science and Drug Discovery Program, a 4 months international intensive industry oriented program to help pharma, life science, engineering graduates to get ready for industry. Once you pass out from college, you have no idea where to go, where to apply, what to apply, but this program will help you to get your job and will help you to make more clear understanding about transcriptomics, genomics, data science stuff, and chemoinformatics. Although this program is for 4 months, wherein a program is divided into 4 different parts. But yes, one can join only for a data science, one can join only for a genomics, one can join only for a transcriptomics, or one can join only for a chemoinformatics. And yes, if you are interested in pursuing your project, final year project with us, you are always welcome to join entire 4 months program, so that we will help you to make a structure of your project, final year project, or we can have a publication out of that, out of this entire exercise. So we usually provide online guidance and offline guidance as well. Till now, we have served more than 3,000 students. We had more than five collaborations. We sponsored several seminar conferences, and we have two coaching centers in India. One is in Chandigarh, one is in Hyderabad. So we have collaborated and acquisitions with several institutions in India like Probiotechnica, Hyderabad Academy, Neogenesis, Anwar Ulum College. And also we are helping several entrepreneurs to grown up in the entrepreneurship journey. And there is a channel called Bows Ahead in the Pharma Toppers YouTube channel as a segment, wherein we usually take interviews of all successful entrepreneurs or budding entrepreneurs from any industry. So as Dr. Mohit said, you will be going through genomics, transcriptomics, data science, along with the chemoinformatics. So if I have to describe about chemoinformatics, it is a field of information technology that uses computer and computer programs to facilitate the collection, storage, analysis, and manipulation of large quantity of chemical data. So chemical data here, it is in sense a drug molecule or a protein molecule. So as most of you might be knowing or might not be knowing, chemoinformatics is a mixture of many scientific areas like informatics, library science, drug discovery, biology, toxicology, text mining, medicine, chemistry, computer science, etc., etc. So yeah, in the entire course duration, you mostly learn about the data science, bioinformatics. Those are the common keywords in the industry, like data science, bioinformatics, chemoinformatics, and drug discovery. So what makes chemoinformatics different from the bioinformatics? So in bioinformatics, you usually learn about biopolymers, sequence of repeating units, but in a chemoinformatics, you usually learn, or mostly you learn about the chemicals, and there is no sequence data and no repeating units. So there is also a different molecular descriptors and comparison methods in between bioinformatics and chemoinformatics. So in the current industry, people are using intensely, they are using data science technique like machine learning, artificial intelligence, natural language processing, computer vision technology in the chemical and biological spaces. So there are several companies in India as well in abroad out of India, and I think there are more than 250 startup companies who are hiring only bioinformaticians and chemoinformaticians, those who know knowledge of chemistry or a biology or a life science or a pharma, along with their data science skills. So now it is a must for everyone to have data science skills along with your domain knowledge called life science, pharma, chemoinformatics, or bioinformatics. So what computer-aided drug design, you might have heard the terminology called CAD. CAD defines computer-aided drug design. Actually, CAD divides into two parts, structure-based and ligand-based drug design. In structure-based drug design, usually we go ahead with a molecular docking. Definitely will, in the entire session, we'll have several hands-on training with a molecular docking in our 4 months training course. And in the ligand-based computer CAD, you will have mostly molecular similarity method, which is usually used for the virtual screening approach. So in the session, we'll have a hands-on session on the virtual screening and how we can convert, how to identify how heat converts into a lead and lead into a drug molecule. So there are some chemical notations like DNA, RNA, we usually do with CG, A, and all this kind of notations with a protein, like with an amino acid. But for chemicals, we don't have any notations or know any sequences. So with the chemicals, we usually represent chemicals with a SMILE structures. So these are the some computer readable formats of a drug called a SMILES or SMARTS or a SMUTS pattern, and also some INCHI and RINCHI formats for the drug. So definitely throughout this course, you will come to know, you will learn about what are the computer readable formats for those drugs, how drug is getting bind to the protein, how protein is reacting in the body to cause any particular side effect or to solve any particular disease condition. And where will bioinformatics and chemoinformatics meet is like epigenomic modification and dealing with the data of chemically modified biopolymers. As we discussed, bioinformatics deals with the biopolymers and chemoinformatics deals with the chemical data. So we can have several sessions on a chemically modified biopolymers, which combines bioinformatics and chemoinformatics, so that we will have broader idea about bioinformatics and chemoinformatics. So I would like to pass on to Ms. Neetu Singh for further discussion on the bioinformatics part and about her company called ProBiotechnica. Neetu, over to you.
Hello, Dr. Sujit. Thank you so much to handing it over to me. Um, Very good evening to everyone. You guys are hearing a lot about bioinformatics, chemoinformatics, and a lot more. So much is heard and delivered to you. So, Sujit, can you please share my slides? I actually want to thank Ilya and Mohit to take me on board in such short notice, and we'll look forward to how it will go. Okay, so Jeet, I will take some time to you to share this. Okay, so we already have talked about so much about bioinformatics and all that. And when I go to the ground level in the college, and when you talk to the students, they have this dilemma that wet lab is, I guess, far more better than the bioinformatics or the dry lab. So there are so many dilemmas still going on the ground, right? So I just want to deal with this very small thing, and everyone already have explained very beautifully and very elaboratively. So I just want to, summarize all the data and everything, whatever we have discussed in this webinar. So, Sujit, can you please share it if you are like hearing me?
I think I'm sharing my screen.
Yeah, but actually I'm not able to see my slide. You can.
Dr. Sujit, we see your slides actually.
Yeah,
is it?
you are sharing
Just give
your
me.
slide. Yeah, yes. Mm-hmm.
If you want me to share, then I can go ahead and also
Yeah,
share.
yeah, sure. Okay, you have to go to the prior slide, Sujit. Yeah.
Yes.
Okay, so yes. My name is Neetu. I have founded and I'm the CEO of ProBiotechnica, and this idea actually came into my mind only and only because I had seen this personally that when we were students, we were facing this problem that where to go for the training, where is the authentic training centers available in the market who are actually, ready to help us to endeavor our future properly. So then I get decided that, okay, fine, I have to find out something where student and faculty and any PhD researcher can blindly, just trust us by our work only. So that is how I just coined this thing. We are just new to the market. We are just 3-4 year old, but still we have collaboration with 5+ colleges. We have taken 30+ training batches. So yeah, we are doing quite well. Sujit, can I see the next slide, please? So I'll give you a very brief introduction of mine. I have done my MTech in biotechnology and gold medalist in that from Amity Institute of Biotechnology, Noida campus. Um, the value which we want to actually add through our services is we don't want to only take care of the student or the faculties or any individual or a group as such, but we actually want to add the value to the institutions, to the faculties, to the industry as well, right? So it's not that just a student is if just a single student is like is very good in one bioinformatics training program. So we are just done with that. We actually want the institutions also to come up and seek for such type of collaboration so that they can, because if you see the market, the students actually believe their faculties. So if their faculties are actually telling them that okay fine, this is something you can, if they pitch them, definitely they trust to somebody who are very new to the field. So we actually want to institutions to come up and to go for such type of collaboration. Sujit, can you please change the slide? You can hear me? Yeah, next one. These are some of our training and works and workshop which we have done. This is what I was talking about. We not only work for the students, we actually want to work with institutions, to the faculties, also in the industry, so that we can actually bridge the gap what the students are studying in the college and what actually the industry is requiring. Sujit, can you go for the next slide? So the question arises, what is difference in bioinformatics in 21st century? Already, Dr. Mohit already has emphasis on this topic. I'll just have a quick, sum up of this thing. Initially, when we talked about bioinformatics, we just simply used to think about this as biology, mathematics, and the knowledge and the complex data, and we have to deal with that. But now the bioinformatics has changed its meaning completely. It's not only the the amalgamation of few domains, but it is like basically analyzing the whole data, which is ultimately going to the betterment of the human being. And the coronavirus is actually the outbreak and the quick remedy of this virus, getting the vaccine just within 1 year. It is a milestone for every life science person. And if you see the global bioinformatics market, the size of this is expected to witness a hike of 19.8%, which is really high by the end of 2023. So yes, as it is discussed earlier as well, that yes, it is booming now. Life sciences, it's not only the life science, it is a blend of bioinformatics, chemoinformatics, and data analysis, and lot more, right? And everyone has seen this thing. So my this is only what we actually have wanted to talk about. Now I'll hand over this to Beeksha to further proceed to the webinar. Thank you so much, everyone.
Thank you so much, Ms. Neetu, for sharing this with us. Now, let me share about the program resources. So what are we going to utilize in this program, in this 4 months of training program, and how it will go? So you already are familiar with the 4 phases of the training program. Now, how do you get started with the training? So I'm going to be introducing you to some of the resources and also answer any question that you may have. So the first resource that we offer is a platform called learn.omicslogic. So learn.omicslogic is our platform for learning. Now, I would like to paste the link to the platform on the chat box. It's a free platform. There are both paid and free resources, but just to get started, you can create an account on learn.omicslogic. I'll also be showing you in a while how to do that. And there are quite a number of free resources that anybody can get started with. And as part of this training program, we'll also be using Slack. So Slack is an app which you can use in your browser or in your mobile. And what we will do is we will have a Slack group for all of our participants spread across the different phases of the program. And you can utilize Slack to directly post any questions that you may have. So these are the first 2 resources that we will use. And the Learn platform is something that I recommend any participant, any faculty, any researcher who is just coming and learning for the first time or joining us here for the first time to get started with. You would notice that in the login section, you have both login via your email ID or any of these social media links. So you can get started and you'll find a number of resources. I will be sharing more about them very, very soon. So moving on, as part of this training and throughout this 4-month program, You will also be guided on how to use the tBioInfo platform. So this is a tBioInfo data analysis server platform. This includes demo pipelines as well as data management and cloud infrastructure to run bioinformatics pipelines. And different stages of analysis are performed in different sections of this multi-omics pipeline. So we are also going to see what this pipeline is all about. Now, Ilya has also demonstrated to you regarding the coding platform. So we have a platform called CodeOmics Logic to train our participants in using R and Python to solve biological questions. So this is a platform which, again, every participant will have access to as part of this 4-month training, whichever phase you are participating in. So our objective with the R and Python platform is not to just train you in either R or Python, but to help you utilize them as tools for biological data analysis. Now, I wanted to highlight, before I go ahead and show you some of these platforms in action, the outcomes that other participants have received. So these are some of these participants sharing about these outcomes from LinkedIn. So some of these participants have worked on a project. For example, Urja, she has worked on a project with us on differential identification of biomarkers between normal neural stem cells and glioblastoma stem cells using RNA-seq analysis, which has been published. She's working towards attending several different seminars and presenting them. So what our objective is to help you get started on a journey. And the project that you do, it's not going to only help you, say, for example, do your maybe the final year project, but something that you can take forward and do more with it, like, even deeper research into this. So we will want to help you get started with that, along with the support of a mentor team. And these are just some of these few examples that I wanted to share with you all as we are getting started. So here are these examples. So now let me quickly take you through some of the different resources right here. So I'm sure that most of you have already registered on this portal. This is very necessary that you're registered here. So if you're registered here, you will receive all the details on how to attend this program. The cost, what are the different breakdown of the costs across the different 4 sessions. So you will come to know about these. And I have also posted the link to this WhatsApp group. So we have this WhatsApp group for all participants that you are welcome to join in. And now let us look at the platforms that I just described. So this is the first platform, again, called Learn.omics Logic. So let us just quickly log in and see. So I'm already, I already have an account here. So you can see that my account is here. I can just sign in. So if you want to go ahead and sign in with me, you can go ahead and do that. And then once you come into the platform, you will notice that there are several different courses. So let us talk about a little bit about genomics, because we have genomics as the first part of the program. So you will notice that once you are inside genomics, you will come across several different lessons which are divided here, and you will be able to take each of these lessons once you're registered. So any lesson that is marked with a star is a premium lesson that is only available to program participants, or if you have a special license. But otherwise, all participants are also welcome to explore some of these free lessons. Like, I would recommend you, if you are going to get started, Introduction to Genomics is a great lesson to get started with. So similarly, you will notice that there are several different courses on different topics as well as projects that you can explore. Now, the other platform that I was talking about is the server tBioInfo platform. Now, I am not going to be showing you a demo of this platform just yet, but just work through the different areas of analysis that you see on the platform. So you see there is an area for transcriptomics, genomics, DNA, RNA, which also has metagenomics included in it. And each time you use this platform, you can highlight on each section of this platform to understand what is it that you are doing. So we will be guiding all participants on using this platform. And the benefit is that you can actually analyze a large data set using this platform. And every participant will have 5 terabytes of space in the cloud to run the different pipelines. So we will be talking more about the platform in our upcoming webinars and sessions, and probably have a demo. So I don't want to spend more time here. The third platform is going to be Code OMICS Logic. So as you see on the Code OMICS Logic platform, we have lessons like how to get started with data science using Python, how to get started with bioinformatics using Python, similarly, how to get started with bioinformatics using R. And the best part is that any participant does not necessarily need to install R or Python on their computer. They can just get started by using the code blocks which will be provided in the courses right here. So these are the 3 platforms that we will be talking about and utilizing as part of this 4-month training. And these would be supplementary to the online sessions that we will be having. So the online sessions would be over Zoom like we are having today, and we will be doing that. So I also wanted to invite you all to go ahead and join our LinkedIn and Facebook groups. So these are just some of the groups that we have on our bioinformatics research community that you can join, and you will be able to see that these groups do not only have students like you, but also has researchers, faculty, administrators who are all working, and even industry specialists who work in this domain. And they will be sharing updates in this group. You will be learning more about job opportunities as well when you're part of either of this group. And we also share all of our videos. Like, for example, this video is currently live on our YouTube channel. So you can also go ahead and follow us on YouTube as well. Now, let me quickly get back to the presentation, because I want to open the session for any questions that you may have, and also ask you all to take part in a poll. So I'm launching a poll on the screen. Now, this poll will help us determine what is the right time to have the maximum number of you be part of a program like this, because we understand a lot of you are having exams. So please participate in the poll. So we have asked you the first question, which is, what are the best months for you to be able to attend this training? Is it from March to June, from April to July, from May to August, or others? If you select others, please put that others down in the chat right now. Okay, Harmin, thank you so much for letting us know. So you can choose a first option, right? Then what time of the day would you prefer to attend this training scheduled? So all of these sessions that Dr. Mohit showed, the 33 sessions, are going to be at around 1 hour session. So we usually have 1 hour sessions like this. It can go to 1 hour, 30 minutes sometimes if the topic is more in depth, but nothing beyond 2 hours. So please let us know. The maximum number of votes will decide, what is the time we should fix for having a training like this. So I see that a lot of you are going for 6 p.m. to 7 p.m. IST. So let us, and also, would you want to attend this full training program? Now, as we mentioned that we also have the training available for you to take in part, and you can either choose genomics, you can either choose transcriptomics or chemoinformatics. That is completely up to you. And what are your research interests? So we also understand that we do have research interests from various people who may be interested, maybe in drug discovery, maybe in infectious diseases or anything else. So please feel free to post it. And if you are writing others, please again post that in the chat box. And how familiar are you with bioinformatics? So if you're a beginner and you do not have an idea of what bioinformatics is, it is all right. We have a lot of these participants. For example, Urja, who you saw doing that project, she came in with not much idea of what bioinformatics is and was able to do a project in a period of 3 months. So it is not impossible at all. These resources, as well as the mentors, would be helping you to achieve that goal that you have of learning bioinformatics and also understanding how to apply it. So we'll teach you the logic and then you apply the logic to your own research projects. So I'll keep this poll on for a few more minutes, and then I'll be closing down the poll. I do hope that we can get a majority vote on this so that we're able to decide on the date and the time, which we will be emailing everyone by next week. So Harmain, you can reach out to us. So my email ID would be marketing@pine.bio. I will put it down in the chat box in case someone needs to reach out to us regarding this program. So please email me regarding any of these questions that you may have. And if you all have any other questions, please ask us right now. So we will be closing this session soon, so please let us know. Okay, so I would be ending the poll right now. I see a lot more of you are voting for the poll. So maybe I'll keep it open and okay. Okay, so let me keep it open for another minute. Yeah, and any questions, again, if you have regarding any of the courses that we shared today, like any of the sessions that we are going to be doing, please ask us and please let us know. And you can save my email ID, marketing@pine.bio. If you have already registered for the program and you have filled out that form, you will receive an email from me, and you will receive quite a number of emails from me. So please make sure that you are saving the email ID. Now I'm ending the poll. And thank you all again for sharing about the intended time and how you would be able to attend these sessions. Thank you all again. So I'll also share the results right now on the screen so that everybody can take a look at this. You are welcome, Nosheen. Great to see that many of you are beginners and would love to learn more. A lot of you are also interested in drug discovery. Then I see infectious diseases. If you want to attend a full training, I understand a lot of you are interested, a lot of you are not. That is great. So I see 6 p.m. to 7 p.m. IST is a good time for the majority of the people here, while May to August seems like the majority for everyone. So thank you all for sharing this. This is really helpful for us. And again, go ahead and follow us on our social media channels. And now I would like to invite Dr. Sujit to share a few words before we end today's session. Dr. Sujit, over to you.
Thank you, Bipsha. Hey guys, very thank you to all of you that you have attended today's webinar. And this is a free webinar which we have conducted. Along with this, we will be conducting one more webinar probably in the next week. We will keep you posted about the dates of the next webinar. And as we have mentioned, we have another collaborator called FABA, Federation of Asian Biotech Association, and the next webinar we will have some great personality from the FABA in the next webinar as well. And along with, as I could see, there are several PhD students, faculty members, masters, and undergrad students. So I would encourage all of you to join this course, either for any of the course or for the entire course. And also, if you are ready to go ahead with your project, final year project proposal with us, along with a paper publication, we'll be happy to serve you for project designing, defining, and doing all such projects in a data science and a biology or chemistry domain. So thank you all of you for your participation, and
Thank you so much, Dr. Sujit. And I want to thank everybody for joining us today. So we'll again send you the update for the upcoming webinar. And please feel free to join our groups, to reach out to us on WhatsApp. We welcome you to ask any questions that you may have. And Have a lovely evening, everyone. Thank you all, and we'll see you all soon. Thank you.